I'll introduce Michelle on her topic. Firstly, Michelle works for Plant and Food Research. She's worked there for about 16 years, mainly in the field of honeybee health, and she had a huge amount of work involved with the Varroa response and years going beyond that. Lately, in the last few years, she's been doing some work towards her PhD. And the topic in her PhD is to do with honey gut bacteria and the role of honey gut bacteria on bee health. So her topic today is New Zealand honeybee gut bacteria has isolation prevented dysbiosis. Now, explain that a little bit. Why the need for this sort of research? It's estimated the annual losses in this area are something like in the area of 10% of hives. We need to understand how to mitigate these losses. Michelle was the first to observe this and to say it is the topic of her PhD, and she'll discuss dysbiosis, which is the change in proportion of gut bacteria. So, welcome Michelle. And New Zealand honeybee gut bacteria has isolation prevented dysbiosis.
Fantastic. Okay. Dysbiosis. What on earth is dysbiosis? Probably the best way to look at dysbiosis is if we can take an understanding from yourselves. So for example, up here we've got, on the left-hand side, we've got a pie chart, and this pie chart has happy bacteria, and that's predominantly what you see in your own gut or in your own mouth, for example. And we have a tiny little bit of bad bacteria up there, but you know, when you're washing your teeth regularly in the morning and at night, you're getting rid of that bad bacteria. And so what's happening is that good bacteria that's supposed to be there will just continue to grow and stay at that level. Dysbiosis is the change that occurs of that unhealthy bacteria. So as the day goes along and you're not brushing your teeth, then the unhealthy bacteria increases. And as you go over to the next day, again, more unhealthy bacteria. And as you continue not to brush your teeth, unhealthy bacteria continues to grow. You start to smell funny. People look at you weirdly. People don't want to sit next to you. So that's the growth of the unhealthy bacteria. So every time you brush your teeth, you are preventing dysbiosis or preventing a change in unhealthy bacteria. So if you can try and think of that throughout the presentation, that will help you to understand what's going on with honeybees. So if I move over to our bacteria, in our honeybees we have 1 billion bacteria. So that's quite a lot of bacteria going on in a single honeybee gut. We see gut bacteria impacting on human health, and we have a good understanding of what is going on in human health in terms of the bacteria that's needed to help digest food, to help prevent pathogens, but we have very limited understanding about what's going on in a honeybee, which is surprising because after humans, it's one of the major animals that has had extensive study on it. So this area is very new to honeybee research, which is exciting because that's what I'm working on for my PhD. Plant and Food Research have given me 5 years to conduct my PhD on a part-time basis, which I'm really enjoying at the moment. So I'm about 2 years in, which is exciting. We see a little bit of bacteria, the importance of bacteria in bumblebees. There is a correlation with bumblebees and bacteria and bumblebee bacteria and pathogens. So when you have particular type of bacteria, then the pathogen decreases. So that's exciting. So it suggests that maybe there is something that's going to pop out of the research in terms of honeybees as well. We look around the world and we hear of all sorts of occurrences, you know, of colonies dying, you know, 35% up there in Alberta, or in Canada. We see other places where they lose 60, 70% of their colonies in localised regions. But here in New Zealand, we look at our graph and our growth of beehives has increased dramatically. We're just tipping the 700,000, which is really exciting for our industry. But they're two very different things. One is looking at colony loss, and the other one is looking at population dynamics. And we tend to get the two confused. Population dynamics is what's going on overall, and overall in New Zealand our population dynamics is increasing, so we're in a good place. But the thing that what's not shown in this graph, this pretty rosy graph, is the colony loss that is occurring on an annual basis. And our best guess of this colony loss, which is related to Varroa, which is related to bad beekeeper management, what was it? PPB? Something like that. Nosema, we've got all sorts of things going on. We see that, and we estimate that to be about 10% loss. Now, on 700,000 hives, that's 70,000 hives that we're losing every year, and I don't like the thought of that. It actually makes me feel quite ill. If we could prevent that or mitigate those 70,000 colonies being lost every year, imagine how this industry would be. That's actually pretty exciting. For you guys as beekeepers, if you could mitigate your losses as you guys increase in your hive numbers, if you could mitigate those losses, that means that your bottom line is obviously going to increase. So that's kind of what stirred my idea, my passion, I guess, for bee research, for bee health research. Like I love the idea that my bees are healthy, they're sustainable, and that I will be able to continue keeping this colony for as long as possible. The idea of losing my colonies is not as exciting. So that's what stirred me on with the bacteria. And what I'd like to take you through is a little bit about what's going on in the gut of the honeybee, and explain that a little bit to you, talk you through about what we already know, and then explain a little bit about what's coming up in terms of my PhD and where I'm heading with that. The factors that are in the bee gut, we've got beneficial bacteria that's present in the bee gut all the time. It's pretty much supposed to be taken up as the bee emerges, and she gets that from her surrounding environment as well as the bees and also the hive equipment that she's on. We have uptake, so we call it uptake because that's everything that goes into her mouth and goes in through the gut. So that's the diet and everything else that she's in association with when she is collecting or foraging. And then the next is pathogens. So these are the ones that come in through the bee gut, and then they gain access to the bee and have an impact on the bee through the bee gut. And foraging is the first part, the uptake part. If we just look at this quickly, we know a lot about this sort of area. Yeah, we know a lot about this sort of area. Whilst she's foraging, we've got food going on, but at the same time, she's also collecting antibiotics. She's also collecting antifungal properties. She's also collecting bacteria. She's also collecting pesticides. So basically, she's not just foraging, but she's actually, well, my supervisor over in the States, he used to call them flying dust mops, but actually, they're more like flying vacuum cleaners. So everything that they touch and they consume is going in through their bee gut, and it's potentially having an impact on them as a bee and then on them as a colony So if we think about this in terms of dysbiosis, what's going on? How do these things that are coming into her gut impacting on the bacteria that's in her gut? Now, we will take antibiotics to kind of help issues that we might have in our bodies, to control bacteria in our bodies. And we look overseas, and everybody is pouring antibiotics into their colonies to control American foulbrood. They are pouring probiotics in there, they're pouring nutritional things in there. Have they really thought about what's actually going into the honeybee and what impact is that having on the gut bacteria? We don't even know what the gut bacteria is useful for, but yet we're prepared to put something into the gut that might actually have an impact on that. So I just want you to kind of keep that in mind. I don't have the answers, but I think that we should be thinking about that. If we're prepared to feed our bees because we think it's going to do better, we might want to find out that it actually is doing better for the bee rather than just putting it in because someone's told us so. So, the uptake, the diet, we know that diet has an impact on pathogens. We know, and I'll talk a little bit about this in a minute, But what we don't know is what's going on with beneficial bacteria and the relationship with diet, and we don't know what's going on in terms of the bacteria and other pathogens. So that's the triangle that I'm trying to untangle for my PhD. What impact does bacteria have on the pathogens? What impact does it have on bee disease? So if we look at diet, we know, there's been some great research, Matilla is one of these ones, that if we look at the diet, we can see that if you feed a larvae protein, or they need protein, sorry. So if we feed a larvae a reduced amount of protein, what happens is we end up with reduced weight of the adult bee. If we feed a reduced amount of protein, then the adult bee doesn't forage as much. It transitions earlier, so it actually goes out to start foraging earlier. So therefore, we know that the age of the bee is clicked from the time that they start foraging, and then they go out and they forage after that point. So if that starts earlier, maybe a week earlier or 2 weeks earlier, maybe they will then die earlier. So therefore, the bee colony itself doesn't live as long, and they start to spiral down. We also start to see, which is very interesting, that bees that are not fed well as larvae end up as adults, and they can't communicate with the rest of their hive very well. So their waggle dance is off. So there's a communication breakdown. So it's not just between humans, it occurs with bees as well. What about adults? So adults, we all know that they need pollen, they need protein, so if they don't get it, they die. That's the ultimate. However, even if they have low levels of pollen or protein, we end up with reduced brood rearing. We end up with worker transitioning earlier. We end up with increase in disease, and we end up with a synergistic effects between disease, so deformed wing virus and nosema. So we've got our bee health here, sort of the area going on, and what we need to know is it the diet itself that is impacting on the bacteria, or is it on the health of the bee, or is it the diet that's impacting on something else within the bee? So we're feeding it, but something within the bee is now being impacted, so therefore that has an impact on the bee health or the colony of the bee. And that's, I guess, what I'm starting to look at. So if we quickly understand what's going on in terms of bacteria, predicted functions of bacteria, we know that there's bacteria already in our bees. And the first thing is we see bacteria in the honeybee guts that metabolize sugars. So that's useful because clearly half their diet is sugar. We see bacteria that are in their guts that break down the pectin. So that's the pollen cell walls, which again is useful to know whether it's real or not, but it's useful to know because it's breaking down the cell walls. And we see other pathogens that are in other insects or in other animals are there and are associated with pathogen defense. So for us, it's really important to understand what's going on with the bacteria. If you guys are super sensitive to gory pictures, I suggest that you look away now. But this is a honeybee gut. So it's opened up and pulled out and extended out onto my lab bench there. And you can see the crop up on the left-hand side and going down through to the stinger on the right-hand side. So it's pretty gross. And I've dissected quite a few of these now, so I'm quite familiar with them. They're quite sensitive little wee things. But my question is, do honeybee gut bacteria protect bees from disease? And that's where I'm moving on. So the first thing that I needed to do was to look to see whereabouts in New Zealand, to look to see if our New Zealand bees were similar to what's overseas that we see in, say, America or some other country. And the reason we needed to do that was because we have a closed population here in New Zealand. We don't allow adult honeybees into New Zealand or semen into New Zealand because of the disease factors that they bring in. And because of this closed population, which we've had basically since the 1950s, potentially the bacteria that's in our honeybee guts could have changed, just like it's changing in your mouth right now. It could also be changing in our bees over time. So what's happened in our time? We don't feed antibiotics here, so that's useful. We're removing bacteria, so to speak. But there are pollens and things that carry antibiotics, antifungal properties, as I was showing before on the foraging page. So there are things that are occurring in the colony that we don't know about. So we analysed, so I took bees from 7 places around New Zealand. I got, there was beekeepers involved and they were amazing. They've provided me with bees. And so what I've done is I've got 21 beekeepers from around New Zealand. Each of those beekeepers have given me bees from 4 colonies, 20 bees from each of 4 colonies. And then these were mushed up, as you can see on the, here, and this Siependorf tube here. And they were then sent off for sequencing, which is exactly what Phil was just showing us earlier on with his sequencing things. And what I found is this. There is one major bacteria in our honeybees, that's the turquoise one down the bottom there, and then there's about 6 other bacteria that play a part, so lactobacillus are playing a part, but the Gillymella one is the one that is the predominant in our honeybees. So the interesting thing about this is, well, how does that compare? How does it relate to what's going on in bacteria overseas? We've got our 7 here, but we have 24 bacterial sort of genus or species that we can identify to. So what's going on overseas? And this is what they have over here. So they have 3 large sort of groups and then a couple of little groups. So theirs looks really different. If I flick back again, you can see our one major one with 6 other groups, and then theirs is 3 major ones. Interestingly, their Gillymella, which is our predominant one, is this one here, the light blue one. So theirs looks in some places like they don't even have Gillymella, and in other places they have more. So what is the change going on? Why is their bacteria different to ours? Does it matter? If it does, what can we do about it? Is it better to have more Gillymella, or is it better to have less? We don't know. Some of the reasons could be because we're isolated, but some of the other reasons could be simply because we don't feed antibiotics here in New Zealand. And, you know, that's predominantly for the AFB pest management plan. However, it's a dangerous thing to be pouring antibiotics into a species if you don't actually know what it does to everything else in there. And so it's probably I think it's a very good thing that we don't pour antibiotics into our colonies, because it doesn't end up in the honey and then it doesn't end up in our bees, causing an impact on our bees. But we don't know the impact of it. And so just like what's going on in your mouth, you know, is it important to kill some of that bacteria or is it not? We don't know. So that's kind of the next step forward. What happens when we pour antibiotics or we feed them particular products? What impact does that have on the bacteria? So the second step that I took then was to look to see what the effect of the factors were on the gut bacteria. So antibiotics and pesticides, diet and pathogens, and how does that impact on our bacterial composition? So the first thing I did was I took the antibiotic that is used worldwide, which is terramycin, to control American foul brood. And I fed colonies, I'm not allowed to feed it into colonies. I got a permit, it took me months. I got a permit, I put my bees and do my bees all in the lab, and then I incinerate them. I'm not allowed to let them out, even though they might be dead. So there is no way that terramycin gets into anywhere in New Zealand through my bees. And then what we did is I found that on the inside the blue circle are these bees that have not been fed terramycin. And they have significantly more than the treatment groups in the red circles that have been fed terramycin. Now just keep in mind this is a log scale, so that means that there's 10 every time increase. So that's not just, just a little bit different. That's actually quite significantly different when you're looking at that graph. That blue graph is definitely different to the red graphs up there. So what impact is this having on our reducing the gut bacteria? We don't know. We don't know which species are impacted. We don't know what the role the impacted species are yet. So that's kind of the next steps for me. What happens when we change this bacteria? What happens to the bacteria? What's the importance of that bacteria in there? So we'll start to look at the link between the bacteria and the pathogens. So what antibiotics and pesticides and diet is having on the composition, but then that then has on pathogens. So if we start to look at, say, I add Nosema to my bees, but they've been fed poorly or they have been, treated with antibiotics, what impact does that have on Nosema? Does that increase or decrease the level of Nosema that's going on in the colony? So I'll choose pathogens that are relevant to New Zealand so that we can start to understand what's going on. So in summary, because my time is almost up and I have to keep to my own timing, I have to kick myself off the stage. The proportions of gut bacteria in New Zealand are different, or it appears to be different from what we find in the USA. We don't know whether dysbiosis has occurred yet, and whether it's because of isolation and whether it's because of the antibiotics that are being fed, but it looks like something's going on there, so it's definitely worth continuing to look at this area. We know that feeding antibiotics reduces gut bacteria, so we know that we need to make sure that what is going into the guts of our honey bees, we need to know what impact it's actually having on those bees. And then also, we need to be aware of what you're actually allowing your bees to be feeding on. What are they feeding on? Do you know what's in the surrounding environment? Do you know what you're putting into them and your pollen patties? What's going on? Make sure that you know so that you can look at what's going on and have an understanding of that. So I'd like to thank all of these people that have contributed to the PhD so far. There's still lots more of exciting research to come, but that's just what I can give you in terms of results at the moment. I've just had a huge amount of support, which is fantastic. So thank you very much. If you've got any questions. Cool.
Yeah, hi. Perhaps it's not a particularly popular sort of research road to go down, but what do you think about the sort of antimicrobials that honeybees are exposed to all the time in New Zealand, such as glyphosate?
What do I think about them, or what do I think might be happening within the bee?
Yep.
It's such a new area, we don't know what's happening. But you can imagine with you put a antibiotic in an area where there is some bacteria, that it's going to have an impact on that. And I expected to see those results with the terramycin. I expected to see the bacteria reduce. But what I was surprised about is that the rest of the world has seen that that's been okay for however many years, 40, 50 years. It's okay to feed bacteria, even though we don't know antibiotics, but we don't know what's going on with that. So I can't give you an answer about the glyphosate, but I suggest and urge us strongly to make sure that we start to look about what's going on in the environment around us and around our bees, and get an understanding of what's going on there. Any other questions? There must be some more. Come on.
Hi.
Yes.
Hi, I just wanted to ask you about the life stage of the bees that you were studying.
Yes.
And how that affected their gut community.
That's a really good question. So these bees that I was looking at, I was emerging them out of their cells. So what is supposed to happen is that when bees come out of their cells, they are supposed to have no bacteria in their gut at all. It is supposed to be azenic. Well, when I did my trials to check to see whether they were azenic and use that as a bioassay, I found that my bees had bacteria, which surprised me because the research says there is no bacteria in the bees and they get it from their surrounding environment. But I think that there might be a little bit in that bacteria, in that gut, that then grows. So we have a colonisation effect going on. So I was using newly emerging worker bees, and then the communities are supposed to be set by age 11 days or 8 days and then 11 days, changing a little bit in their proportions, but it's supposed to be set at sort of 8 days. So the initial work is to look to see what happens on these newly emerging workers, because that's what a little bit of work has been done on already, and then move on to see how that impacts on adult bees and how colonies that are actually already sick, whether that has an impact on the colonisation of the bacteria as they grow. So, so we have a sick colony here, and then all of these adult bees are all sick, and then we put in our newly emerged bees. What happens to those bees? Do they get a sick bacterial composition, or they're fine? So it's another interesting question. Thank you.
Yeah, just did you do any studies regarding hives that were under organic treatment or under chemical treatments in regard to the bacterial count inside of the bees?
No, we haven't done that yet at all, but it's an interesting thing, and I'll write it down. Yeah, thank you. Yeah.
That was a great talk. Thanks, Michelle. It's great that you're.
Oh, sorry. I was like, whoa.
It's great you're actually looking into that. It just can't be a real easy one to check out. I wouldn't have thought the little tiny bee guts. But what made you go to America as opposed to anywhere else in the world for that?
But just because that's the first area of research that's been done in this area, the larger kind of area of research that's been done. A woman called Nancy Moran is running a group there, and she's looked at other animals and then has transferred that down to honeybees. So that's why I particularly chose comparing it with Moran's papers and Moran's work, and she's given me her sequencing data to be able to compare that. So we're working through that process at the moment, which is really intense, and it's not that pleasurable for me, but I'm pushing through.
No, first, fair enough. And have you thought about, because it's so close, checking out Australia, and because they haven't been affected by Varroa yet, and seeing if that might be...
Exactly, yeah. I think that those sorts of things are definitely questions that I really want to be able to look at some point. So I've only got 5 years for the PhD, so I'm going to achieve what I can in these 5 years, and then I have the next 20, 30 years to figure these things out.
Good luck with it.
Thank you.
I was looking and wondering about the link between the mycorrhizal fungi that the bees eat at times, and the link between that and the gut health.
That's a good question. What we find is that most of the bacteria that is in the honeybee gut is not found elsewhere. It's not in plants, it's not really. There is a little bit coming in, but it's the community that's in the honeybee colony and in the honeybee gut is very different to what you'd find on flowers. So I don't think there's an impact of the actual bacteria coming in from what's in the environment on the gut bacteria, but I do think that there's an impact, I'm thinking that there might be an impact of what is in the environment, what they're feeding on, that might change their pH in the gut, so therefore that would change the bacteria, or there might be something else going on that has an impact on the gut and the gut bacteria. So yeah, not so much about what's in the environment that's going to impact on the gut bacteria in terms of not so much of what's the bacteria in the environment, but what other parts of the environment will have an impact on the bacteria. Does that make sense? Thank you. Cool. Okay, well, thank you for your questions, and I'll see you in a minute.